Oil flowers and oil-collecting bees, Malpighiaceae and their pollinators. In large areas of South America, as here in the state of Sao Paulo, only remnants of the original primary forest are left. However, these areas are also still very rich in species today. The crowns of tall trees project above several lower stories of woody plants. In this richly structured vegetation, woody lianas use freestanding trees as supports to reach the vital sunlight. Members of the Malpighiaceae are among them. In this family, approximately 950 species are known alone in the neotropics. In the Brazilian Mata de Santa Genebra Nature Reserve near Campinas, most species of the Malpighiaceae start flowering in February, including Stigmaphyllon lalandianum, whose densely foliaged festoons are now covered with a profusion of light yellow flowers. This species belongs to the group of the oil flowers, a type of blossoms that have developed a most unusual and fascinating relationship with their pollinators. The flowers are completely devoid of nectar, but yet they are pollinated by bees that are attracted by a special fatty oil. The bees selectively search for this oil. Relatively few genera of bees share this peculiarity. They belong in part to the solitary Centridini, a tribe of the family Apidae. These oil-collecting bees vary considerably in their body size. The bumblebee-sized, somewhat ponderous species, as here Centris colaris, prefer to fly along the periphery of the liana curtain in their movement from flower to flower. However, the smaller species are much more versatile flyers and manage to reach even the flowers deeply hidden among the foliage by hectic flight maneuvers. At least 8 different species exploit Stigmaphyllon's oil. This tireless collecting activity has a single purpose: to provide food for the bees' offspring. It does not serve as food for the adults themselves. Only the females care for the brood. Therefore, the females alone engage in gathering food for their larvae. The males merely play a role as sexual partners. During oil collecting, the animal's ventral thorax becomes dusted with pollen. This pollen adheres to the stigmas of other flowers during subsequent visits. A flower reaches approximately 15 millimeters in size. There are 5 long-stalked petals. The middle, superior petal, termed the vexillum, provides the bee with a foothold. The back of the flower bears the oval oil glands or elaiophores, which are arranged in pairs as special appendages of the calyx segments. 4 twin glands alternate with the petals, but the lowermost, median pair is absent. In the course of evolution, the elaiophores have most probably originated from extrafloral nectaries, which are still present on the green leaves of the plant. These glands are epithelial elaiophores. That means that the oil is secreted by a palisade-like epidermis. This secretion lies concealed beneath a cuticular blister. Only after pressure has been exerted by a stroking bee leg does the cuticle burst at a preformed weak spot and an oil drop oozes out. After the flower bud has opened, the calyx segments with the elaiophores bend forward directly between the claws of the petals and thus become easily accessible to the bees. The densely arranged reproductive organs of the flower are difficult to differentiate visually. 2 whorls of 5 stamens each make up the androecium. The gynoecium has 3 stylodia and stigmas. Even on the buds, the elaiophores are clearly visible. They are already filled with oil the day before blossoming. Anthesis of a single blossom lasts 2 to 3 days. A bee, here for example Centris scolaris, is seen harvesting the oil with her fore and middle legs. The yield is transferred to the plumose trousers of the posterior legs and is finally unloaded into the brood cells as a larval food. These storage and transporting organs are called scopae. Fourfold slow motion allows observation of how a bee approaches a flower with her antennae stretched forward. Due to the bee's weight, the flower drops a considerable distance. The oil is removed from each twin gland by 3 scraping strokes of the legs on average. The bee alights and tightly grasps the claw of the vexillum with her mandibles. The collected oil initially remains in the densely pilose areas of the fore and middle legs. This scanning micrograph shows the foreleg of a Centris species. The oil collectors are located on the metatarsus of the front and middle legs. From there, the collected oil seeps into the plumate fringe on the outer flank, visible above, where it is temporarily stored. A comb of elastic spatulate chitinic setae comprises an oil scraper. 3 large broad setae are located opposite the comb. As seen here, these are absent from the middle legs. This bee performs a purely oil-collecting flight, as is indicated by her moist, darkly shiny scopae. The liquid is passed from the forelegs to the middle legs by squeezing it out between the bent legs and is finally pasted onto the inner flanks of the posterior legs. Here again, the oil is squeezed out and then backloaded onto the scopae of the hind legs. Epicharis rustica, a somewhat smaller species of the same affinity, also displays a corresponding collecting behavior. Her scopae are yellowish in color and carry a mixture of oil and pollen. Her leg pairs scrape alternately to the right and then to the left, etc., across the elaiophores. Although flower buds are also intermittently visited, enough pollen adheres to the bee. To demonstrate this, a female was immobilized. Her ventral thorax is heavily powdered with pollen. The scopae conspicuously contrast to this due to their fox-red coloration. Their special coloring probably plays a role as a cue in their mating ritual. Note these animals' long tongues. In this slow-motion sequence, all steps of oil collecting and backloading can be observed once again. Positioning, grasping the vexillum and landing, oil scraping, backloading. The bee spreads her scopae backward to secure the load. To recover from this energy-consuming collecting flights, the bees exploit sources of nectar to nourish themselves, such as this Arabidea species of the Bignoniaceae. She spreads the oil from her forelegs to the inner flanks of her scopae. In this action, Epicharis does not differ from the other Centridini. Once again, the backloading procedure. It can be regarded as modified grooming behavior. To keep her legs available for pollen transfer to the scopae, the bee hangs onto a leaf by clinging to it temporarily with her mandibles. Here an Epicharis female is grooming her body. Despite her small body size of only 15 millimeters, Centris similis is also a legitimate stigmaphyllon pollinator. Compared to the larger species, however, she is capable of more versatile flight maneuvers. While orienting herself above a flower, here in slow motion, the bee can obviously check the latter's oil content. Perhaps her antennae determine whether it will be worthwhile to exploit the flower's oil based on the latter's odor. Here the backloading procedure can be observed 3 times in sequence. Centris varia, the tiniest species observed. She performs especially hectic flight maneuvers. She also transfers the yield to her scopae with similar skimming movements, and occasionally spreads her hind legs upward. Centris varia prefers to visit the buds of the flowers, perhaps because the open flowers have already been emptied as a result of the strong competition by larger species. Because the vixillum is not yet available for clinging, she must use her posterior legs for support on landing. Since the petals do not yet obstruct our view, the scraping of the elaiophores can easily be observed. These oil-collecting bees differ considerably in their choice of breeding sites. Some Centris species make use of tunnels burrowed by coleopteran larvae in old wooden posts, but most species dig their own burrows for rearing their larvae. About 110 miles west of Campinas, there is an experimental station of the University of Ribeirão Preto. Here, the breeding behavior of wood-nesting Centris species is investigated in detail. To this end, short pieces of hollow bamboo cane are offered to the bees as a nesting aid and are readily accepted by them. Here a Centris vittata female has chosen her breeding place. She constructs a series of brood cells in the tube by applying a mortar-like mass of oil and sand grains. An occupied piece of bamboo has been split lengthwise. This allows us to look at the nest. A layer of dried oil covers the outer wall that consists of stabilized sand. The wall's upper half has been removed cautiously to be able to look into the brood cells. In the youngest brood cell, a larva is floating in a nutrient paste of oil and pollen. The larvae of the following older cells are, with one exception, more mature. The original primary cell is located at the posterior end of the tube. Its larva will be the first to pupate. The cell's heterochronous establishment makes pupal stages of differing lengths necessary. Only in this way can the youngest individual in the foremost cell hatch first and to leave the nest through the entrance. There are residual primary forest areas near the experimental station. The Malpighiaceae Banisteriopsis muricata grows in this area. Its luxuriant inflorescences glow in rose and white colors. A horizontal axis bears flowers of different ages. During anthesis, their colors change from crimson to white. The reproductive parts of the Banisteriopsis flower resemble those of stigmaphyllon. However, in this case, they are divided into 2 functionally different complexes. The one directed downwards provides fodder pollen for the bee and its young. This complex consists of 1 style and 8 stamens, 5 of which bear large glands. These glands produce an oil that possibly enables the pollen to better adhere to the bee. The second complex is the reproductive one. It is formed by one pair of upright, long-stalked stamens positioned on either side of the vexillum and each accompanied by a style with a small stigma. The Banisteriopsis flower also exhibits 4 narrow twin oil glands when viewed from the back. They are absent on the 5th median calyx segment. The notch on the sturdy vexillum claw has dark necrotic scars. They reveal that it has already been grasped by bees. These sequences, taken at normal speed, illustrate how zealously and thoroughly the bee works in inflorescence. Although Banisteriopsis is visited by other species of Centridini, the details of the collecting procedure will not be discussed further. The oil harvesting procedure is the same in all these species. It has only recently been discovered that flowers offering fatty oil to pollinators, although they represent a minority compared to the classical rewards of nectar and pollen, nevertheless play an important role in certain plant communities, both in terms of ecology and in number. Apart from the extraordinary collecting behavior shared by these bees, the coincidence of distribution areas of both partners reflects their coevolution. Besides the Malpighiaceae, pollinator attraction by floral oil has also evolved in other, taxonomically very distant families. This allows the conclusion that we are dealing with a strategy that has independently evolved several times.